What up? It's your boy, Mr. Jennings here. I'm about to give you some biology. We're going to talk about enzymes today, and that was too cringy. Alright, it's Mr. Jennings. We're going to talk about enzymes because we start Unit 3, Cellular Energetics, and enzymes are a key critical understanding, not just for energetics, but for biology as a whole. Alright, that's enough interlude here. This is Mr. Jennings. This is Big Orange Biology, and class is in session. Now, enzymes are your biological catalysts. Most of us know them as proteins, but they can also be RNA. We'll talk more about that much further down the road, but RNA can do the same things. Now, enzymes have the ability to facilitate chemical reactions. What that means is they are a catalyst, and that means they increase the rate of reaction without ever being consumed in the reaction itself. They do this by reducing the activation energy of a chemical reaction. Now, they do not alter the free energy available. Now, free energy is the amount of energy necessary for reactions to take place. It's in the surroundings. We'll discuss more about free energy in a future video, but because they are a catalyst, they do not alter the amount of free energy. They do not absorb or release anymore. Now, biological catalysts like enzymes are required for almost every biological reaction, and they are highly specific in the process, meaning there are thousands upon thousands of different enzymes in the cell, each targeting a very specific substrate. And all along here, they control the reactions of life. Now, substrates are the reactants that bind to the enzyme. So you're going to hear me use that word substrate quite a bit, especially in this unit, amongst others. And you need to understand what that is. Now, the enzyme substrate complex is what we call the association between the enzyme and the substrate when they're temporarily bound together. The reaction is going to result in a product. We call that, well, the product of the reaction. It's the end result here. And then there's the active site. Now, the active site is where the main reaction takes place. Is the enzyme's catalytic site. Is where actually the substrate is going to bind into it. And see there, it fits into it because it is highly specific to its shape. Now, enzymes are highly reaction specific because each enzyme only works on a very specific substrate. The chemical fit between active site and substrate is going to be through hydrogen bonding and ionic bonding and some covalency here and there. But typically it's a temporary weak bond. Now, overall, this is not going to be consumed in the reaction because, again, that is the definition of a catalyst, something not to be consumed in reaction. Every single enzyme can actually catalyze thousands or more reactions per second. They do this at a tremendous speed. Now, enzymes are going to be unaffected by the reaction itself. However, cellular and environmental conditions like temperature, pH, and salinity have an impact on it because enzymes mostly are proteins. Now, enzymes are named after the reactions they catalyze. For instance, sucrase breaks down sucrose. Proteases break down proteins. Lipases break down lipids. And DNA polymerases build DNA. They add nucleotides to the strand. And pepsin breaks down protein, specifically polypeptides, the more diminutive form of the protein. Now, typically we viewed these models we used to view enzymes back in the early aughts when I was in high school. We called it the lock and key model. It's very simplistic. The idea that the substrate is like a key and it's highly specific to a very specific lock. That would be the enzyme. And when they get in there, the enzyme fits in there, holds the key just like a lock and key. It's great. It's beautiful. It's outdated. The induced fit model is the more preferred model these days. That is more accurate because it shows the 3D structure of the enzyme contorts and changes its shape, leading to a much tighter fit, creating what's called a conformational change between the enzyme when it binds. Because this conformational change, it actually causes the substrate itself to do whatever the reaction needs to be, and when that reaction finishes, it's no longer the same substrate. It then falls off the active site and allows the enzyme to return back to its normal form. So a variety of mechanisms help lower activation energy. Now, some enzymes are synthetic. They have active sites that orient substrates to each other in the correct position. It basically allows for the mechanisms to take place that are necessary. Basically, they bring things closer together and then make them react. And some are digestion. The active sites, whenever they do the conformational change, will put stress on the bonds and physically rip it apart, much like a log of wood. So you have synthesis and digestion. We have what's called anabolism and catabolism. So we're going to talk more about that in future videos. Now, to wrap things up here, today was meant to be a short video. Proteins are not the only types of enzymes, but enzymes themselves are very important. They help speed up chemical reactions. They're not consumed, and there's the bell, but they're not consumed, and As a process, they are repeatable. They're highly specific to a very specific substrate. All right, next time we're going to talk about how does these various environmental factors affect the protein and how the actual enzyme does the reaction it does. That said, this has been Mr. Jennings. Thanks for watching. Have a great day.